Hello and welcome to nucleotide biosynthesis. Let's see purine biosynthesis today. Purines, the nucleotides are adenine, guanine, thymine, cytosine, and uracil, which are divided into purines and pyrimidines. Purines are adenine and guanine. Today we are going to deal with the purine biosynthesis. The purine biosynthesis can be done in two pathways. One is the de novo synthesis. Another pathway is salvage pathway. In the de novo synthesis, purine ring is synthesized from small molecules, all new molecules, whereas the salvage pathway, it is nothing but that it is synthesized from the degraded nucleotides. Recycling of the purines is done in the salvage pathway. Coming to the de novo synthesis, here it refers to the synthesis of complex molecule from simple sugars such as sugars and amino acids. That means the purine ring is derived from amino acids as well as some other sugars like ribose sugar and one or few atoms which are attached to the ribose ring, and it is not synthesized from the free bases. It is synthesized from the free bases in the salvage pathway, whereas in de novo pathway, all these adenine and guanine, they are synthesized from small molecules and they are not synthesized from the recycled products. Whatever the digested nucleotides, they are also converted into waste molecule, degraded into uric acid, but not utilized here. Let's see, this is the skeleton of the purine ring, which has 9 atoms. Among them, this 4, 5, 7 molecules are donated by glycine, 8th one is donated by formate, and this 3rd and 9th nitrogens, they are donated by the glutamine amino acid, and this first nitrogen is donated by aspargine molecule, and this carbon C2 and 8, they are given by formate. This 6th position carbon is given by the carbon dioxide, respiratory carbon dioxide. And this pathway, which one? The salvage pathway as well as the de novo pathway, they begins with the phosphoribosyl pyrophosphate. At the 5th position, the phosphate group is attached. First position is occupied by pyrophosphate. It is 5-phosphoribosyl 1-pyrophosphate. This is the basic structure on which the salvage pathway and de novo pathway are dependent. This is the entire pathway of the purine biosynthesis. It results in the formation of inosine monophosphate. From the inosine monophosphate, the nucleotides ATP and AMP and GMP are constructed in just 1 or 2 steps. Let us see, this is comprised of only 10 steps, and the first one, this is the preliminary step, the basic step. Here, alpha D ribose 5-phosphate, it is converted. Alpha D ribose 5-phosphate, it is converted to PRPP, which is nothing but the 0 step or the preliminary step both for the de novo and salvage pathway. Here, this is in the alpha and it is also in the alpha PRPP, and ATP molecule is converted into AMP, donating its 2 phosphates to be attached to the first carbon. PRPP is synthesized by phosphoribosyl pyrophosphate synthase. From the PRPP in the first step, now we are starting the first step. PRPP is converted into 5-phosphobeta-D ribosyl amine. Here only the amino group is added. That amino group, here this is the amino group which is donated by the glutamine. The amino group donor glutamine, it is converted into glutamate. Here the pyrophosphate, these 2 phosphates are released in the form of pyrophosphate. This pyrophosphate, it drives the molecule to be converted from alpha to beta by utilizing the energy. Later on, this 5-phospho-beta ribosylamine is converted into glycinamide ribosyl 5-phosphate. Here, glycine molecule NH2CH2COO- is nothing but glycine. These three molecules are added. Glycine donates three atoms, as we have seen. These three atoms are added to the nitrogen amino group of on the ribose 5-phosphate. Thereby, it forms glycinamide ribosyl 5-phosphate. Very simple. Glycinamide ribosyl 5-phosphate. This glycinamide ribosyl 5-phosphate, it is later converted into formyl glycinamide. That means the formyl group. Formyl, this is the formyl group. Formyl group is added to the glycine, thereby forming formyl glycinamide. Formyl glycinamide, later it is converted into formyl glycinamidin. Glycinamidin means to the glycine. Here this amino group is added. That is why it is called as formyl glycinamidin ribosyl 5-phosphate. We will see the enzymes later. Here, this amide group is donated by glutamine, where it is converted into glutamic acid. After this one, this formyl glycinamidin is converted into aminoimidazole by the ring closure. The ring is closed between the 8th and 9th position, resulting in the formation of imidazole ring, to which the amino group is attached. That is why it is called as aminoimidazole ribosyl 5-phosphate. This aminoimidazole ribosyl 5-phosphate is converted into aminoimidazole carboxylate. That means carboxylate group is added. Where it is added? It is added at this one. At this carbon, the carboxyl group is the carboxyl group donor is nothing but carbon dioxide. This carbon dioxide is added to the 5th position. Here, this is the carbon dioxide, resulting in the formation of aminoimidazole carboxylate. Later on, it is converted into aminoimidazole succinyl carboxamide. Succinyl carboxamide, it is carboxylate here and converted into carboxamide. So it is COO here, it is converted into CONH. That is why carboxamide. This nitrogen NH group donor is nothing but aspartate. This amino group is attached to the carboxyl group here, converting it to carboxamide. Later, this one aminoimidazole succinyl carboxamide ribonucleotide is converted into aminoimidazole carboxamide ribosyl 5-phosphate. Aminoimidazole. The formic acid, fumarate, it is broken down from this one. It is cleaved. The aspartate is broken down at this position, forming fumarate. The amino group is attached, forming aminoimidazole carboxamide ribonucleotide. This one, the formate group is added to this nitrogen. In the next step, formate group is added to this nitrogen. So to the nitrogen, formate group is added. The donor is a formyl tetrahydrofolate, forming formimidazole carboxamide ribonucleotide. Formate imidazole group. Later on, in the last step, it is converted into inosine monophosphate, where the ring is closed between this nitrogen and this carbon, resulting in the formation of inosine monophosphate. The inosine monophosphate is the basic step, where it is utilized both for the synthesis of the adenine as well as AMP as well as GMP. IMP is the basic compound from which AMP and GMP are formed. In all the reactions, only 10 reactions are there. This is nothing but the zero reaction, synthesis of pyrophosphate, and among them, this is the first step is nothing but the rate limiting step, where the synthesis depends on this reaction, whether the nucleotides or it has to be synthesized or not, depending on the availability of the PRPP molecules. And the conversion of PRPP to phosphobeta deribosylamine is the rate limiting step. Usually it is inhibited by AMP and GMP. Means Why means when AMP and GMP are abundant, they inhibit the first reaction. When AMP and GMP are abundant, they inhibit this particular enzyme, thereby the products are not synthesized. That means inosine monophosphate is not synthesized because there are abundant AMP and GMP molecule. And azacitidine is a glutamine antagonist which acts as an anti-cancer drug, which inhibits the reactions involving glutamine. The glutamine here, the glutamine is involved, and another stage here also the glutamine is involved. These two stages are inhibited by azacitidine, which is used in the cancer therapy. This is nothing but the simplified steps of the purine biosynthesis, de novo biosynthesis. Ribose 5-phosphate is converted to phosphoribosyl PRPP. The enzyme is PRPP synthase. Here you can see the enzymes. PRPP is converted into phosphoribosylamine. The enzyme is PRPP glutamyl aminotransferase. Phosphoribosylamine is converted to glycinamide ribosyl 5-phosphate. Just amide group is at glycine is added here. And this enzyme is GAR synthetase, glycinamino ribosyl ribonucleotide synthetase. Glycinamidine ribosyl 5-phosphate is converted into formyl glycinamide ribosyl 5-phosphate, and the enzyme is formyl glycinamido amino ribosyl aminotransferase enzyme. It is converted into formyl glycinamide ribosyl 5-phosphate. The same is repeated here, which is converted to amino imidazole ribonucleotide. You can pause this and you can see the reactions carefully, then you can observe very well. As the time is approaching, I am going faster, but you can stay on this slide and you can observe carefully. Amino imidazole carboxylate ribonucleotide is converted to amino imidazole succinyl. So succinate group is added here. It is donated by the aspartate for the sake of amino group, and this is converted to amino imidazole carboxamide ribonucleotide, which uses the fumarate molecule is released. After this, it is converted to this molecule. After adding formyl group, it is converted to formamino imidazole carboxamide ribonucleotide, which is later converted in the last step. Cycle is ring is closed, forming inosine monophosphate. And inosine monophosphate is the basic molecule, as I told you. It is used for the synthesis of both AMP as well as GMP. Here, inosine monophosphate, simply it is converted to adenylosuccinate, where the molecule is donated by aspartate CH2, COO, CHNH3, COO minus, where the amino group here, it is donated to the inosine monophosphate, forming adenylosuccinate. The molecule is still attached. When the fumaric acid is released, the amino group is attached, resulting in the formation of adenosine monophosphate, AMP. So AMP is synthesized from the inosine monophosphate by the enzyme adenylosuccinate synthase enzyme. And here the inosine monophosphate in the next pathway, it is converted into GMP, guanosine monophosphate, in the two-step process. Inosine monophosphate is first oxidized to xanthosine monophosphate, whereas the NAD is reduced. The enzyme is IMP dehydrogenase, inosine monophosphate dehydrogenase. The IMP is oxidized to xanthosine. The oxidized xanthosine, here you can see the oxidized xanthosine, it is converted to guanosine monophosphate by the transfer of amino group from glutamine to the xanthine monophosphate, forming the guanosine monophosphate. The glutamine is converted to glutamate with the help of the enzyme transaminidase enzyme. So this process of purine biosynthesis results in the formation of the main compound IMP, inosine monophosphate, from which adenosine monophosphate and guanosine monophosphate are synthesized. These two are the purine nucleotides. The Adenosine monophosphates are converted into diphosphates and triphosphates. Nucleotide monophosphate is converted into nucleotide diphosphate by phosphoribosyl kinase, as well as it is converted into triphosphate by the monophosphate kinase and diphosphate kinase. And let us sum up this one. The stages, this is the basic reaction which you have to understand. When you understand this, you can remember very well the purine biosynthesis.